Hello, everyone. To those in Facebook and to those who join us in Zoom, welcome to our first Research Insider session. So this event is co-organized by Malaysian bioscience scholars, which is MBios, 100 centers in Malaysia as well, who just came on board a few days ago. They also have a lot of cool scientists that we will be speaking to. And we also brought in Science Media Center Malaysia, who is our media partner. So let's welcome Wen Wen.
Okay, so I think you guys should be able to see my screen now. So yeah, like Apple mentioned, my name is Wen Wen. I'm a second-year microbiology PhD student based at Imperial College London. So for those of you who just joined, like my virtual background is actually a picture of one of the labs at Imperial from the building where I work in. So this is where I work, what it looks like. And today I'm going to tell you guys a little bit more about myself, my research area, and what I do in the lab. So I thought I'd start off by telling you how I ended up doing a PhD in microbiology. So talking a bit about my education background. So even as a kid growing up, I would say that science was definitely my favorite subject at school. And I definitely have a lot of really great teachers to thank for that. But also my parents were very supportive of it. So they let me watch a lot of TV, but that TV included like Animal Planet and Discovery Channel. So I think that definitely helped. And when I was younger, they also bought me lots of those like illustrated children's encyclopedias. And I fondly remember one of them that I read so much that the spine was a little bit like tattered. I've since given it to my niece because I want to, motivate her to have that similar interest in science. So it was, I guess, all bred from a very young age. But in terms of where I studied, I only studied in Malaysia up to Form 3 actually, because when I was 15, I received something called the ASEAN Scholarship. So for the next 4 years of my education, I was actually living in Singapore. So I did my O-levels there. That's the equivalent of the SPM. And as you can imagine, I was in the science stream. But then after that, when I moved on to do my A-levels, I had to decide between whether I liked biology or physics more, because that's just the nature of like the Singapore A-levels is you have to pick between one of those two to get in the science stream. And even though I used to do better actually in like physics exams, I knew that my interest was in biology. So I ended up just doing biology anyway at A-levels. While I was there, I think that interest really grew in me as well. So I decided that I was gonna probably do like a biosciences related degree at university. So in terms of extracurriculars, I really tried to pursue stuff that I thought would boost my application. So I did a small research project in the lab where I learned a lot of molecular biology techniques. And that's a picture of me in the lab with a bacteriophage soft toy on my head. And I also trained as part of the school's biology Olympiad team. The Singapore Biology Olympiad exam is probably the most difficult test I've ever taken in my life. But thankfully, it didn't discourage me or demotivate me enough because I still wanted to do a biosciences degree at uni. So I applied to several universities in the US and the UK, but I eventually decided to accept the offer to do biochemistry at Imperial College London. And I spent my undergrad years, I think, going through what most undergrads go through, which is, trying to figure out what I want to do afterwards, what kind of career I want to go into and things like that. I only really decided that I wanted to continue to pursue research in final year, which is actually pretty late because I didn't manage to get any research attachments or internships the years before. So the internships that I did were all not science related, but I really missed the science. Like I missed doing something that was related to what I was studying. But in final year, I also started taking modules that I was a lot more interested in. So I think by that time, I had figured out that I'm really interested in infectious diseases, and I took modules in microbiology and parasitology, and that really like solidified that interest for me. So, that was when I was starting to consider pursuing a research career, studying like infectious disease biology. But I didn't have, as I said, the experience. So I didn't want to commit to like applying for a PhD straight away. And my personal tutor at university, she advised me to apply for a master's degree instead to try and gain that experience. So I did a 1 year's research master's degree also at Imperial, where I did 3 lab rotations. So my first rotation, I actually worked with parasites, called a parasite called Toxoplasma gondii. You don't need to know what that means. My second project, I worked with enterohemorrhagic E. coli, which if you've read the news, it's the bacteria that causes these like really bad cases of food poisoning in the US that are linked to contaminated lettuce. So that's what I worked with for my second project. And in my final project, that's when I started working with Klebsiella. And that was actually with to do PhD where I'm at right now. It was a great project and I really liked the people that I was working with. So when the principal investigator, my supervisor, offered me a PhD, I said yes. So what do I actually work on? For my PhD, I'm trying to understand how antibiotic resistance spreads between bacteria. I think everyone knows what an antibiotic is. You've probably been prescribed one at least once in your life. So antibiotics are drugs which kill or prevent the growth of bacteria, and specifically bacteria, so they're not effective against viruses or parasites. They belong to like a much larger group of compounds that we call antimicrobials, and these are just things that target all microorganisms, but antibiotic specific to bacteria. And the reason they're specific to bacteria is because they target processes that are only found in bacteria. So these processes can include the production of the bacteria cell wall, which is the structure that surrounds it and sort of protects it from the environment. It also targets like processes like how the bacteria produces its genetic material, so DNA and RNA, and also its proteins. So all of these processes are very essential for the bacteria to survive. And if we block them, then the bacteria will die. So I work with an antibiotic called a carbapenem, and that antibiotic actually targets the cell wall and prevents it from being built in the bacteria. So the first antibiotic ever discovered by Sir Alexander Fleming is called penicillin, and that antibiotic also blocks the production of the cell wall. So I guess you could say what I'm working on is like a modern day version of that first ever antibiotic that was discovered. Now, unfortunately, and you may have heard, but bacteria are better scientists than we are. So whatever we are trying to attack them with, they are starting to become resistant to them. And this is a really big issue, obviously, because if we can't treat these infections, then people are more likely to die from them. And bacteria can become resistant to antibiotics by several different strategies. So they can actually prevent the antibiotic from getting into themselves. They can also produce these like little enzymes, which I've just represented with this like Pac-Man thing here, that can destroy antibiotics. If any antibiotics have gotten in, like some bacteria are able to actually make pumps that can actually remove them straight back out of the cell. And lastly, they can also change the target that the antibiotic is trying to attack. So when the drug gets in, it doesn't recognize anymore what it's supposed to be attacking, and it's just ineffective. The bacteria that I work with, Klebsiella pneumoniae, it's a really common cause of hospital-acquired infections. So these are infections that people would get when they're staying in the hospital, usually for other reasons like maybe cancer treatment or surgeries. And these bacteria are starting to show a lot of resistance to those carbapenems that I mentioned previously, especially by these first two strategies. So they are very good at blocking the carbapenems from getting in, and they also make little enzymes that can destroy any carbapenems that have entered. And in my area, like what I'm researching specifically, because I'm trying to understand how resistance can spread, it's especially relevant to this second strategy here, because bacteria basically, they need genetic instructions in order to make these enzymes that can destroy antibiotics. And what they can do is they can actually share those genetic instructions with other bacteria. So I've just like illustrated that over here. So these genetic instructions for making these antibiotic destroying enzymes are usually encoded on this small piece of DNA, this little circle that I've drawn over here, that's called a plasmid. And basically, these plasmids can move between bacteria. So a bacteria that we call a donor can actually donate this plasmid to another bacteria that doesn't have it. And then now that bacteria has the instructions that it needs to make these enzymes. So basically now both bacteria are resistant, and that's how they actually share and spread resistance. So this process is called bacterial conjugation, and that's the basis of my PhD, is trying to understand the mechanism behind the process so that we can try and inhibit it or block it to try and slow the spread of resistance. And this kind of research is really important because we are rapidly heading towards what I think we can only describe as a huge AMR crisis. So as I mentioned before, antibiotics are a type of antimicrobial. And antimicrobial is a really big issue because currently it's estimated that infections caused by antimicrobial resistant microorganisms cause about maybe 1 million deaths per year. But by the year 2050, which is in 30 years time, we think that number will get up to 10 million people per year. And by then, it's going to kill more people a year than cancer. So it's going to be the major killer of people in hospitals, especially. And one of the reasons, like, this is going to be a big issue, we have bacteria like the one that I'm studying is really so WHO has even highlighted Klebsiella, which belong to this group of bacteria here called Enterobacteriaceae, and especially the ones that are resistant to these carbapenems. They've classed it as a critical priority pathogen for developing new antibiotics, because essentially these bacteria are resistant to almost everything that we have. And that means that we can't treat them with anything. So the other reason why, it's becoming such a huge issue, this resistance thing, is because we are running out of antibiotics. As I said before, bacteria are becoming more and more resistant to everything that we have, but we're also not really discovering new antibiotics to try and combat them, the resistant ones. And one of the reasons for that is usually antibiotics are actually found in nature. So like I mentioned, like penicillin, the first ever antibiotic discovered, it was actually found just by random chance. Like a mold was growing on a plate of bacteria and it was killing the bacteria, and they discovered that it was creating an antibiotic. So that's actually generally how we find antibiotics, is we look to nature to see what kind of compounds they're making that can kill bacteria. But I guess in the same way that, over time, these resources are finite, just like oil and gas. We can keep mining them forever, but they will sooner or later run out. So we are starting to, we're basically not able to find as many antibiotics as before. So the number that we are releasing every year is going down as we speak. So research like mine is quite important because we want to, buy time for scientists to look for more antibiotics, as well as to come up with new strategies against antibiotic-resistant bacteria. And we're buying time basically by trying to lengthen the amount of time that we can use the existing arsenal of drugs that we have. So trying to limit the spread of resistance actually is really important in helping them, just have a little bit more time to find these kinds of things. So that's like the whole research area. I think now I'll just move on to sort of what I do on a daily basis, so you can figure out what I do in the lab. So here I'm just going to describe like a typical day in the lab. And the reason why I put typical in like quotation marks is because I would say every day in the lab is slightly different. I don't think there's such a day, like no day is the same basically. And that is, for me, the exciting thing about being a scientist, but also the very challenging thing about being a scientist, is you have to be very flexible and you have to adapt to, unexpected things happening that can totally throw all your plans out the window. But a typical day, and this is probably the most common type of day for me, is, I get in the morning and one of the first things I do is I check to see whether my experiments from the day before have actually yielded any kind of results. So I work with bacteria. So bacteria grow on these things called, like we grow them on Petri dishes that we fill with agar. So it's just got all the nutrients that the bacteria need to grow. And so I just check to see, have they grown? How many of them have grown? Do they look slightly different? And things like that. So that's like the results from my experiment, is looking through all these Petri dishes. I also set up experiments in the lab. So I think in the preview post for this session, I mentioned like one of my most important pieces of equipment is actually a Bunsen burner, because I do everything sterile and we have to make sure that nothing contaminates our media. In the morning, I also like have a lot of meetings with people. My lab is quite a big lab. So sometimes I need to have like smaller group meetings just with the people that I work with my day-to-day supervisor, so we can, discuss plans and things like that. Towards like afternoon time is when I might, sit down on my computer, respond to emails, do a bit of writing sometimes, prepare presentations, because as a scientist, it's not just about doing experiments, you also have to communicate your research. So it's really important for us to also, take some time to actually present our data and like figure out how to make sure that other people are aware of what we are doing. I also do a lot of molecular biology. So for my work, it involves creating a lot of mutants. So I do a lot of gene editing in bacteria. And one of the main processes involved in all of that is actually using PCR, or polymerase chain reaction. So this machine here is a PCR machine. And I've just put this picture here because this was from a time where I actually hogged every single PCR machine in the lab. So that's just, to show you how much I use this process in my day-to-day work. And I also do a lot of just like replenishing resources. So making petri dishes, making up new antibiotics, setting up bacterial cultures. So I have the stuff that I need for my experiments. Towards evening time, when it gets a little bit more quiet, that's when I'm winding up experiments. You know, I check to see whether my PCRs have worked. These gels don't always come out so nice. And then during that time, the office also gets a little bit more quiet. So I do a bit of reading. I catch up on the latest research and literature. And then I also do a lot of planning, because I think as a PhD student, you're expected to be quite independent in your research. So it's really important just to make sure that you're organized and you know what is coming up in the next few days or the next week or so. And lastly, I was just going to tell you a bit about some of the techniques that I tend to use in the lab. As a microbiologist, obviously, I do a lot of microbiology, so I characterize bacteria quite a bit. One of the ways we can characterize them is actually measure how they grow. So this graph here actually just shows like 2 mutant strains of bacteria. The one in black is actually growing slightly better, so this is just over time. And you can see it's getting to higher numbers earlier, so that just means it's growing better. And this green one just doesn't grow as well. We can also check to see how they react with different antibiotics. So we just call this resistance profiling. All the sort of creamy areas you see here, that's all bacteria. And these like blue areas are actually where they've not managed to grow, because these little pieces of paper here actually contain antibiotics, and they can diffuse into the agar, and everywhere that they've diffused to, if that bacteria is sensitive, so if it's not resistant, it will just die. So that's actually how you can measure if a bacteria is resistant or not to a specific antibiotic. And lastly, I do quite a bit of microscopy to try and look at bacteria at the cellular level. So this is a picture showing Klebsiella that I've tagged with different fluorescent proteins so that you can actually see the different individuals in that population. So I don't know if you can see, but there's some that are not colored, so they're gray in color, and then there's some that are red, and there's some that are green. So if I have a little bit of time, I'll explain to you more like what that experiment is going on. But I basically use microscopy to see how resistance is spreading in a population. And as I said, like I do a lot of molecular biology. Those fluorescent bacteria don't make themselves, we have to make them. And that's where all like PCR and mutagenesis comes in. So that's about it in terms of what I've prepared.
Yeah, so actually, Wen Wen, we have a question that came in from Tom. So he asked, are there any strategies for combating antibiotic-resistant bacteria besides discovering new antibiotics? Because you know, we're running out of antibiotics, so we need new ideas. Can you share with us some?
Yeah, definitely. So personally, if you ask me, I think this, you know, trying to look for new antibiotics and then just like throwing them at bacteria is not a very sustainable cycle, because they just become resistant to them eventually. Bacteria are much better scientists than we are ever going to be. So again, it's not sustainable. You know, we find something new, we put it on the bacteria, they will become resistant to it, like inevitably. So that's why a lot of people have been coming up with new strategies. I think one of the most ideal things that we could do is actually to come up with a vaccine for bacteria. But historically, it's been very complicated to vaccinate against bacteria as compared to viruses. We've had a lot more success with vaccinating against viruses than bacteria. I think the most famous like vaccine against bacteria would be the one for TB. So the BCG vaccine, I think everyone has gotten as a child. If you check your shoulder, that little mark there, that's your BCG vaccine. So tuberculosis is caused by a bacteria, but even that vaccine is not like 100% effective. Like people can still get TB even though they have been vaccinated. So I think, yeah, vaccines against bacteria are just a lot more complicated, but there are other strategies as well. So there's phage therapy. It's very experimental at the moment, but you know how we have viruses? I mean, everyone knows that there is a virus right now causing a pandemic, but viruses can also attack bacteria. So there are specific viruses that only attack bacteria, and those are called phages. So actually, there's a picture.
Yes, it's called bacteriophage. So
yeah,
viruses
yeah. So
that
it attacks bacteria specifically. So actually, this is a soft toy of a bacteriophage that's on my head. Yeah, so it's a virus that attacks bacteria specifically, and like people have been using it in cases where bacteria are multi-drug resistant. So, you know, if any drug doesn't work anymore, we're at last resort now. So what do we do? So actually people have given patients phages to try and destroy the bacteria using a virus. It has worked, but I think it's also complicated because bacteria can also become resistant to phages. The way that bacteria can become resistant to phages, I think now is very well known because everyone has heard of our most famous gene editing technology, CRISPR. So CRISPR is actually the immune system of the bacteria that against phages. CRISPR is developed from the system that was used by bacteria to actually fight off phages. But actually one of the most interesting concepts that I've heard recently, and we had this really interesting seminar by a professor from the US who came to speak at Imperial, is actually not to attack the bacteria, it's to promote tolerance. So if we don't kill the bacteria, we don't encourage resistance. Her concept is if we can try and make the immune system tolerate the infection without killing the bacteria, then we're not going to encourage resistance. And that's, to me, is a really interesting concept. So
how do you do that?
So one of the ways that they found, so their experiments are really quite interesting because in a lot of animal experiments, for example, or just experiments in general, if you give a specific dose of bacteria to a population, you can measure something called a survival curve. Like 50% of the population would die, but 50% would survive. Like with any infection, not everyone dies, right?
Yeah.
So is actually trying to figure out what is special about those people who survive, and can we give that, you know, that factor or whatever to whoever is not going to have as good of an outcome. So that was really, really interesting to me.
Just a quick question to the audience. Would you accept bacteriophage therapy if we run out of antibiotics to cure your infection if you have one?
Also, Julian's question is about what's more important, like supervisor, team versus a research project. I think obviously a best case scenario is that all of those are good. You know, you have a good team, you have a good research project, etc. For me, actually, I think I am in that kind of ideal situation. I personally think I have a really great team, and my project is also very interesting. Yeah, but I think overall, I would actually probably say that the people are more important.
Not your PI, is it?
Well, the PI as well, but I mean, everyone, you know, in general that you're working with, is the environment. I think the environment, if you're working with people who are very encouraging, I think even if your project may not be the thing that gets you out in the morning, but at least, you know, there are people who are supporting you. I think that's very important. Like,
and also, like, my experience, like when you go into a lab, your PI is so busy that he can't answer your questions. Like, it's your friends who help you out, you know? It's your lab mates.
Yeah. So everyone, like the whole environment needs to be very supportive, honestly.
What softwares do you use for your work?
So actually, my work is very, very basic analysis. Like, it's not like very complex data analysis compared to a lot of other people in my lab. I basically just use Excel.
Excel.
When it comes to like statistics and stuff, then I use like GraphPad, Prism. I think that's what most people use as well. But other than that, when it comes to designing mutants, I'll just do like basic multiple sequence alignments and like blasts. It's not anything too complicated because all of my work is really just like counting, putting numbers into Excel.
There are experimental evolution experiments to use bacteria to design antibiotics themselves.
So definitely, a lot of the antibiotics that we use today are actually found in soil bacteria because the soil, sort of the bacterial biome in the soil, you know, that it's constant warfare basically. So, you know, all the bacteria in the soil are just making antibiotics to try and like fight each other, as well as resistance mechanisms to try and fight each other. Actually, one of the other reasons why it's been so difficult for us to find new antibiotics is because a lot of these bacteria, like in the soil, for example, we can't grow them in the lab. That is one of the big issues. So one of the teams in Imperial actually is really cool. They're coming up with a platform to try and grow bacteria that you otherwise can't grow in the lab by sort of like mimicking those conditions. So they're almost trying to like mimic soil in some way using like some kind of platform that they've designed, because the idea is we are still not able to find a lot of these things simply because they cannot be isolated in the lab. So we are definitely looking at bacteria, for example, to find new antibiotics. I think that definitely is something that has been looked at for a while, yeah.
Because when one makes mutant bacteria, so how you make a mutant bacteria is you delete the gene that you want to study in. Wen Wen has this very interesting question for you. How so how long do you guys think it takes to generate these mutants?
Because when I talk to my friends who are not in science, like they always think that it's a very quick process. So I was just wondering, like in general, what do you all think? Oh, by the way, like my friend made me this like toy version of one of my mutant bacteria. I don't know if
Oh,
you
it
can
has
see
hands. Oh, is that
No,
hands?
these are the things that it uses to transfer DNA. Yeah.
Oh. Oh, okay.
Oh, okay.
Some people think it's 1 hour. Actually,
I wish it took 1 hour.
if it took 1 hour, science would like just skyrocket, and science productivity would just skyrocket. But what's the answer, Wen Wen? How long does it take for you?
So, assuming everything goes all right, and this is actually a very big assuming, is because, you know, obviously a lot of things can go wrong along the way, but if all the PCRs work and everything comes out great, I think maybe around 2 weeks.
What about anti-virulence therapy? This way you don't kill the bacteria, but instead you target the virulence factor. So virulence factor is basically proteins that are infectious, that
make
help,
you sick.
that help with the infection. So what is your thought about anti-virulence versus antibiotics? And maybe a combination of these two might help. Does it speed up the evolution of resistance or not?
So actually, I think that's a really good point. I guess a combinatorial therapy like you suggested is a really good idea. Obviously, I think the thing that we're trying to stop is we don't necessarily want to kill the bacteria. We just don't want them to cause us disease. And that's where the virulence factors come in, right? So we have obviously been exploring, the use of, trying to stop those disease-causing processes instead. But oftentimes, like these disease-causing processes are also the same processes that the bacteria sort of need in order to survive within us. It's multifactorial, basically. Like the whole thing is part of a very complex process. So obviously, while that is, a nice idea, I don't know of anyone who's managed to do it successfully. Mentioned before, like my second project, I was working with enterohemorrhagic E. coli, and we were actually, looking at a virulence factor and trying to see what role that is playing in the host, for example. But I don't really know how exactly we might be able to use an anti-virulence target against it. I think because the disease-causing process, I think, is very, very complex. It usually involves many different things. And for you to target all those different things, it's going to involve coming up with lots of different compounds, potentially.
Which technique do you use for gene deletion?
Oh, okay. So I actually do what we call like scarless recombination using a Gibson assembly. Just like recombination, so it's just homologous recombination to knock the gene out.
Would like to know how you secured your funding for the PhD.
Oh, okay. That's a bit of a complicated thing because like when it comes to my funding situation is not very conventional, which is why I didn't bring it up. It's a bit of like a weird agreement between like my department, my supervisor, and stuff like that. Yeah, so it's not like conventional funding.
So is it funded by the uni or?
Sort of, like it's like a scheme thing, like a bursary scheme thing that the department came up with.
And how do you target the genes to knock out? Is it by comparative genomics or transposon mutagenesis?
When I make my mutants, everything is targeted. So it's definitely like I do the bioinformatics first. I figure out which genes actually look like good targets to knock out. So I've not done transposon mutagenesis, that actually means that you're just like randomly targeting genes and trying to see like which one has a function.
What is the latest manipulation of microbiology in the biotech world?
Oh, okay. I'm not a biotechnologist, so I can't tell you like what's the latest that's going on there. But I think you've heard of microbes that can eat plastic now.
I know, and, exactly. It's a very in thing.
Microbes that can produce oil as well.
And like microbes that also clean up the environment. That's called bioremediation. If you guys have heard of it.
It's really terrible. They can cause disease and stuff, but also I think they are a big part of potentially what we can consider like a circular economy if we know how to use them. Yeah.
What is the general environment like for women in the microbiology field?
Oh, okay, that's a good question. So Microbiology is an interesting one because we are actually very highly represented by women in this field. There's a lot of women in microbiology. In my lab itself, I mentioned it's quite a big lab. So my PI is a man, but out of like 16, 17 of us, I think like there are 13 ladies.
Wow.
There's only like 2-3
Girls
guys
represent.
in the whole lab. Yeah. And every time he advertises for a position, like we always get women applicants. That being said, I think there is obviously still a little bit of inequality. You know, just because there's a lot of women doesn't mean that you still don't get women not getting grants, for example, or women not being promoted to like higher position. And it still has to go back to all of those, like, you know, women want to start a family, they might not be seen as like ideal candidates for becoming a PI or a lecturer or things like that. So, you know, even though there's a lot of representation, which I think is really great, but I think more can be done in terms of equality.
What is the best and worst thing about working in a wet lab?
So I think best thing, I think personally, I just prefer that kind of style of doing experiments because I like doing things with my hands and stuff. Like I'm an arts and craft person and I love to cook as well. So to me, it's like being in the lab is a bit like doing my hobby all day long. Like I procrastinate from writing by going into the lab and doing stuff. But the thing I don't like, especially about microbiology labs, is because a lot of the work that we do has to be sterile. We use a lot of disposable items, so there's so much waste.
Plastic.
Yeah, so much plastic waste.
Guys.
And I feel like, you know, we as people who are aware of the problems with climate change, but we are also contributing to that. It's painful for us, like trust us, like we don't want to do it, but you know, because we want our experiments to work, and it will actually be more wasteful if, you know, we didn't use these kinds of products, for example. Yeah.
One last question from Julian. What are your plans after your PhD?
Oh, okay. So I'm still like mulling on that at the moment. Obviously now this like whole pandemic has made things a little bit uncertain about when I would even finish because our universities are all thinking about extensions and things. But, you know, I am not too sure about staying in academia just because I think the thing that I like most about science is getting to do the research, and once you get into academia and you go higher and higher up, that's when, you know, you start having to just like fight for money and things like that, and I don't really know if that's what I want to do. I like the teaching aspect of it. Like I think it would be great to be a lecturer, but usually in universities you're expected to be a lecturer and a researcher, you know, combined.
At the same time.
So one of the other options that I had been thinking about is going into industry, so potentially like pharmaceuticals, and hopefully in something that's related to antimicrobial resistance research or something, you know, trying to find new strategies against this issue.
Okay, so for more updates, you know, like our Facebook page on Unbiased, on 100 Scientists of Malaysia. And, you know, thank you again for your support and coming in on a Saturday night. And hope to see you next Saturday as well, where we will chat with Alea, who is investigating how learning and decision-making are affected in teenager OCD. It's called obsessive compulsive disorder. Yep, we'll see you guys next Saturday, and thanks for joining me and Wen Wen today.
Yeah, thanks for hosting.
See you guys. Yeah, bye.